Welcome to this lecture on signal transduction pathways. Today, we are going to explore the fascinating microscopic communication networks that keep our bodies functioning. We will uncover how a tiny molecule on the outside of a cell can trigger a massive, life-saving response on the inside. This topic is absolutely foundational to human physiology, and by the end of this session, you will understand both the biology and the mathematics behind how our cells talk to each other. Now that we know what we are studying, let us start with a simple analogy. Imagine you are a delivery driver trying to drop off an urgent package at a highly secure factory. You cannot go inside, so you press the intercom button at the gate. The guard radios the floor manager, who then starts the assembly line. In the human body, this exact scenario happens millions of times a second. The hormone is the delivery driver, the cell membrane is the secure wall, and the receptor is the intercom button. Because many hormones cannot enter the cell directly, they must bind to the outside and trigger a chain reaction of messengers inside. This internal relay is what we call a signal transduction pathway. Moving from our analogy to the formal biology, let us define what signal transduction actually is. In physiological terms, it is the biochemical process by which an extracellular signaling molecule, like a hormone or neurotransmitter, activates a specific membrane receptor. When the hormone binds, it induces a conformational change in the receptor shape. This physical change is crucial because it activates intracellular proteins that generate molecules called second messengers, such as cyclic AMP or calcium ions. These second messengers then activate specific enzymes, usually protein kinases, which attach phosphate groups to other proteins, ultimately altering the overall activity and behavior of the cell. Building on that definition, it is important to understand that not all hormones deliver their messages in the exact same way. Peptide hormones are water-soluble, meaning they cannot cross the fatty lipid bilayer of the cell membrane. Therefore, they must bind to surface receptors, like G-protein-coupled receptors, to initiate the internal cascade we just discussed. Conversely, steroid hormones have a different chemical structure. They are lipid-soluble, which allows them to bypass the cell membrane entirely. Instead of using surface receptors, steroid hormones slip right into the cell and bind to intracellular receptors, directly altering gene transcription in the nucleus. Now that we understand how the signal gets inside, you might wonder why the body uses such a complicated, multi-step relay system instead of a direct mechanism. The primary reason is a powerful phenomenon called signal amplification. This is absolutely critical for human survival and efficiency. A single molecule of adrenaline binding to a heart cell receptor can trigger the production of thousands of second messengers. These, in turn, activate millions of enzymes, causing your heart to beat faster in a fraction of a second. Without this amplification, your body would need to produce impossibly massive amounts of hormones to get any physiological response at all. Beyond just amplifying the signal, this multi-step process serves another vital purpose. These pathways provide multiple checkpoints for regulation. Having several steps allows the body to fine-tune its responses, turn them off quickly when the danger has passed, or integrate signals from different hormones simultaneously. It is a highly controlled system. However, if these pathways break down or mutate, the consequences are severe. For example, in diabetes, insulin signals are ignored by the cell, leading to high blood sugar. In cancer, cellular growth signals might be permanently turned on, causing uncontrolled cell division. Proper regulation is a matter of life and death. Before any of this amplification or regulation can happen, the initial connection must be made. The hormone, also known as the ligand, must successfully bind to its receptor on the cell surface. We can understand this interaction using the principles of chemical equilibrium and molecular kinetics. The likelihood of binding depends on the concentration of the free hormone, the concentration of the free receptors, and how strongly they are attracted to each other. This attraction is quantified by a specific value called the dissociation constant. It is important to note that a lower dissociation constant actually means a higher affinity, meaning the hormone binds very tightly and easily to the receptor. This concept of binding affinity leads us directly to the mathematical model that describes it. We can calculate the exact fraction of total receptors that are currently bound to a hormone using the fractional occupancy equation. Let us look at the symbols. The symbol Y represents the fractional occupancy, which is the proportion of total receptors bound to the ligand, ranging from 0 to 1. The symbol L in brackets represents the concentration of the free ligand floating outside the cell. Finally, Kd represents the equilibrium dissociation constant we just discussed. Conceptually, this formula shows that if the ligand concentration increases, the fractional occupancy Y also increases, meaning more receptors are activated and the cellular response becomes stronger. Now that we have our formula, let us apply it to a real physiological scenario involving insulin. Suppose a muscle cell has insulin receptors with a dissociation constant of 2 nanomolar. After a large meal, the concentration of insulin in the fluid surrounding the cell rises to 6 nanomolar. We want to find out what percentage of receptors are bound. We start by substituting our given values into the equation, placing 6 in the numerator for the ligand concentration and 2 plus 6 in the denominator. Simplifying this gives us 6 divided by 8, which equals 0.75. Physically, this means 75% of the insulin receptors are currently bound, indicating a very strong signal is being sent to absorb glucose. With the receptors successfully occupied, we can now calculate the massive internal cascade that follows. To find the total amplification, we need to multiply the number of molecules produced or activated at each step of the pathway. This formula captures the cascading nature of the pathway, where every step multiplies the effect of the previous step. Our total represents the final number of altered target proteins that will actually do the work inside the cell. Ng is the amplification at the membrane level by G proteins. Nc is the amplification by the second messenger generator, cyclic AMP. And Np is the amplification by the final kinase enzymes. If any of these variables increase, the final physiological response grows exponentially. Let us see this incredible power of signal amplification in action using the fight-or-flight hormone, epinephrine. We are given that one single active receptor activates 100 G proteins. Each of those leads to the production of 1,000 cyclic AMP molecules. Finally, each kinase phosphorylates 500 target proteins. We want to calculate the total number of phosphorylated proteins resulting from just one single hormone molecule. We substitute our values into the amplification equation, multiplying 100 by 1,000 and then by 500. The final calculation reveals a staggering 50 million phosphorylated target proteins. Physically, this massive number illustrates how a tiny, almost undetectable amount of hormone can instantly prepare your body to run from danger. To solidify our understanding of these calculations, let us look at a visual representation of the entire cascade.
In this diagram, you can see the cell membrane separating the outside environment from the inside. At the top, the first messenger, or ligand, binds to the G protein coupled receptor, or GPCR. Notice how this binding triggers the G alpha subunit to detach and move along the membrane to activate adenylate cyclase. This enzyme then produces the small circular molecules you see labeled as cyclic AMP, our second messengers. Finally, these second messengers travel down to activate protein kinase A. This image perfectly captures the physical relay race that our mathematical formulas just described. We have covered a lot of ground today, so let us summarize the most important ideas. First, signal transduction pathways are the essential communication networks that allow cells to perceive and respond to their environment. Second, we learned that the initial step, receptor binding, is a precise mathematical relationship driven by ligand concentration and affinity. Third, by utilizing second messengers and enzymatic cascades, the body achieves massive signal amplification and strict regulation. Whether it is insulin lowering your blood sugar or adrenaline preparing you for action, you now understand the biology and the math behind the microscopic relay races that keep us alive every single day.